So, good morning, everyone. We are going to continue our journey for Patterns of Inheritance, Chapter 10. So, last time we talked about monohybrid cross, and then I told you guys Gregor Mendel, he was the father of genetics. He's the one who figured out the traits get passed on and the Punnett square. So, this is called the Mendelian genetics. We talked about dominant alleles or recessive alleles. And then this is where we stopped. So we stopped right here. Last time I said that you guys have the maximum in this class will go up to is 2 genes. So monohybrid cross mono means 1. You're looking at 1 gene and it's going to have 4 squares. Whereas dihybrid, that's what we're going to talk about today. Dihybrid cross has 16 squares. Di means 2, and we are looking at 2 genes in this case. So 2 genes are represented with different letters. In this case, you have R and G. That gives you 2 different genes. If it was uppercase R and lowercase r, it's still the same gene, it's just a different allele. It's a different version of the gene. But when you have 2 different letters, that means you have 2 genes. So if you have heterozygous parents for 2 genes, you're going to do this cross. In this case, you still have 2 genes, but you don't really have heterozygous. It's most likely homozygous recessive and then homozygous dominant. So you don't really need to do the 16 square. You can still do 4. Sometimes students just do 16 because it makes a little bit more sense to them. But this can be done and you'll just say 100% yellow. And then we are meeting for the lab today and we will do some problems for that. But just to remind you guys, let me show you what monohybrid is and then we'll go ahead and do an example of a dihybrid cross. So for this example, I'm going to say, so let's do monohybrid first. I'm gonna say big T is tall and little t is short. And the question is cross 2 heterozygous parents. And first question is ratio, genotypic ratio. So what's the genotypic ratio? Genotype ratio. And then the second question is to find out phenotype ratio. So that's the question. So first thing you want to do, you want to write the genotypes of the parents. Help me out here and type them in the chat. What are the genotypes of parents? Big T, little t, yes. So it's big T and little t for 1 parent, but we said we crossed 2 heterozygous, so big T, little t again. What are the, not the alleles, what are the gametes? What are the gametes for this? So one possibility is big T and the other is little t. So these are the gametes. If this is male, I could, draw a tail and say this is a sperm. And then here, same thing. The possibility is big T and then little t. So those are the gametes. You split them in the gametes after meiosis. So these are different possibilities of gametes. Now you go ahead and you do the Punnett square. You put big T, little t, big T, little t. You bring this big T down, this one across, same thing, this one down, this one down, this one across, and then little t, little t. So that's your Punnett square. And this generation, this is called F1 generation, first generation. F1 doesn't stand for first, it stands for filial, which means family, but you can try to remember it as first generation. So these are the parents. These were the parents. When you cross the parents, it gives you F1. So I'm gonna say when you cross the parents, it's going to give you F1 generation, the first generation. If you were to take this F1 generation and cross it with another F1 generation, when you cross that, it's going to give you F2 generation. If this was in humans, this would be incest, but we're talking about plants here. So this is going to give you F2 generation. Now going back to this question, what is the genotype ratio? If you look at this Punnett square, what is the answer here? Please type it in the chat. 3-1, 1-1, okay. So when you write the ratio, I want you to also tell me what is 1, what is 2, what is 1. Yes, Kaylin. So this is the genotype ratio. Truce, I think you might have written the phenotype ratio. And then 1 little t, little t. That's what you got here. Now, for what is the ratio for phenotype? And write it as you would answer it. So what is the ratio for phenotype? What are the traits we are looking at? So I want you to go back here. So 3 tall and 1 short, yes. So this is what we are looking at, traits. And then your phenotype is 3 tall and 1 short. Just to make sure, is this making sense to everybody? This is monohybrid cross. This is an example of monohybrid. I'm gonna compare it to a dihybrid next. Please let me know yes, no, any questions on the monohybrid cross. Otherwise, I will move on to dihybrid now. Any questions? Please let me know in the chat. No? Okay. So now let's do a dihybrid. So let's do a dihybrid here. So for dihybrid, I said it means 2 genes. So we are going to look at 2 genes. And just for this example, I must say one gene is A and the other gene is B. Those are the genes. What are the possible gametes? What are the possible gametes for this gene and then for this gene? Again, type it in the chat. What might be the possible gametes for this gene? Yes. So if we big A, little a for this one, and then big B, little b for this one. So this is one gene. This is one gene. This is another gene. Let's say second gene. This is an allele. Little b is allele. Big B is also an allele. Same thing here. Big A is an allele. Little a is also an allele. I'm not gonna write that, but that's also an allele. So these are alleles, and these together is a gene. So that's the gene. Now, to get the possible gametes, so we just got the genes. Now to get the possible gametes, we're going to use something that's called FOIL method. I don't know if you guys used it in algebra. Have you guys heard about FOIL method before?
Yes, it is being used only on when you are working on Dihybrid.
Yeah, so this means first, outer, inner, last. So first, outer, inner, last. So these are our genes. I'm gonna write that again. So we're gonna apply this FOIL method to this. So to get the gametes, this is going to give us the gametes. So first, we're going to do the first letters, big A, big B. Those are the first ones. What are the outer? What are the outer?
Small a, small b.
Small a, small b. No, actually, it's going to be big A. This is the outer one, and this is the outer one. So let me redo that one. It's going to be big A, little b. Then inner. Now you have little a and big B. And then last is little a, little b. Those are your gametes. So in this question, I gave you these genes. These are heterozygous, and I could say this mates with heterozygous female. So same thing for the female. Now you could, you don't need to redo this because you already know this is what you're gonna get. And you could do 16 squares. You could put these 4 on top, and then the same thing on the side because you're mating with the same parent with the same alleles or heterozygous parents, just like the male parent here. So you could just put those down here. You don't need to do a separate FOIL method because you have the same alleles here. If it was different, then you would do it again. And then you just do the cross. I'm not gonna do this. I have an example on the slide. I'm gonna show you that. But this is how you set up a dihybrid cross. And let me show you the example. So this here, they just have different letters, but it's still heterozygous. Female gametes heterozygous, big R, little r, and big G, little g. Male gametes also heterozygous, big R, little r, big G, little g. If you do the FOIL method on this is what you're gonna get. If you do the FOIL method on this is what you're gonna get. Then you do your Punnett square. And there's very important value that I want you to remember, and that is 9331. Anytime you take a heterozygous female gamete for the dihybrid and heterozygous male gametes for dihybrid cross, you're always going to get this ratio, 9331. I might not have you do heterozygous dihybrid cross for the exam, but I want you to remember this ratio. So I might just ask you, what is the ratio you would get if you made it heterozygous male with the female for dihybrid? Then you're gonna say it's gonna be 9331 ratio. So this is a very important number, write it down.